My name is Camille McCall. I'm a postdoctoral research associate at Rice University working on the Houston Wastewater Surveillance Monitoring Program. In this module, I'll be discussing quality assurance and control for quantification of microbial targets in wastewater. This is the second module in this series on quantification of microbial targets. To understand the steps and methods involved in microbial quantification, you can refer to the first module in this series entitled Methods for RNA and DNA Quantification of Microbial Targets in Wastewater. I first want to start with discussing quality assurance and quality control as it pertains to wastewater surveillance. QA, or quality assurance, primarily focuses on the processes and techniques used to improve the quality of WBE programs, such as training, contamination control practices, and processes to ensure the results are accurate, processes to optimize target recovery, and strategies used to efficiently scale up when needed also fall under the QA. QC, or quality control, focuses on the results or the output of the process. It consists of identifying and tracking contamination based on process controls, methods for validating target concentration, such as consistency among replicates, meeting PCR thresholds, and its consistency with peer-reviewed literature. This module will cover a mixture of QA/QC topics with a focus on contamination, process controls, and target recovery. There are three main learning objectives for this module. We will discuss possible sources of contamination and what measures and practices can be implemented to mitigate those sources of contamination. Secondly, you will gain an understanding of what positive and negative process controls are, how they are used, and where they can be implemented in the wet lab process. And lastly, I will discuss target recovery, identify stages where loss can occur in the laboratory process, and ways to minimize loss and improve recovery. In the next 3 slides, I'm going to cover common contamination issues, ways to reduce contamination, and an example from our lab of showing measures that we put into place to control for contamination. Sample contamination can be defined as the introduction of any undesired impurity, such as chemical, microbial, or physical matter, into a sample during sample collection or any stage in the quantification process. Specifically, in a laboratory setting, contamination can occur as a result of poor lab hygiene practices, such as improper use of personal protective equipment like lab coats, gloves, and face coverings. Poor maintenance or cleaning of equipment and workspaces, and improper sterilization of consumables are also poor lab hygiene practices that can lead to contamination. The way a lab space is constructed can also contribute to contamination. In this case, contamination can occur when there are overlapping work and storage spaces for different stages of the quantification process. Poor communication between working groups can also play a role in contamination. For example, failing to communicate when equipment was last calibrated or cleaned, or poor labeling of reagents and samples. Lastly, human practices such as poor pipetting techniques and sample handling can lead to cross-contamination. Several practices can be implemented to reduce contamination. These include routine cleaning of workstations and equipment, proper use of PPE, and appropriately sterilizing consumables. It is also best practice to designate rooms or spaces, equipment, and storage areas for each stage in the quantification process to reduce contamination. Airflow controls, such as positively and negatively pressurized rooms, can also be implemented to reduce contamination between rooms and spaces. Lab construction will depend largely on the space and funds available and should be designed accordingly. Automating steps of the process can greatly reduce contamination from human error and can increase workflow efficiency. Ultimately, whether automated machines are used or not, all lab personnel should be trained on not only how to perform each step, but also best practices in sample handling and pipetting techniques. As an example, This diagram shows an early layout of 2 out of 3 spaces that we have designated for our WBE surveillance program. Our sample receiving and concentration room is designed to meet BSL 2+ standards, and it has its own equipment, PPE, and storage. Next to the concentration room, we have our nucleic acid extraction room, which contains its own equipment as well. Separate PPE is used by technicians for each step in each designated room. For this next part of the module, I'll be discussing positive and negative process controls. Much of the information I'm going to share is based on the detailed recommendations found in the EMMI guidelines paper shown here. Process controls are critical to validating experimental designs and answer 2 main questions. Is the assay working, and is there contamination? Positive controls let us know whether the assay is working or not. Here I'll be discussing 2 types of positive controls: internal and external standards. The internal standard is usually of a different target than your target of interest and is added directly into the wastewater sample to provide some type of information about the sample, whether it is how the composition of the sample impacts recovery, or even how much human input is contributing to the sample. Internal standards can also help to identify variations in the process and aid in estimating recovery. Specifically, if the target of interest cannot be easily obtained, a surrogate microbe can be used. Examples of internal positive controls include surrogate microbes and human fecal indicators. As opposed to internal standards, external positive standards are usually the same microbe as the target of interest and are added to a non-sample matrix such as DI water. External standards can also be added directly to the sample, but the background concentration of the target in the sample is usually below the limit of detection, has already been measured and accounted for, or is being measured in parallel to the spike sample. Using external standards can show expected results, and similar to internal standards, they can aid in evaluating the process and determining target recovery. Negative controls, opposite to positive controls, are samples that should be free from microbial activity or at least not contain any detectable concentration of the target of interest. These controls therefore help us to answer the question of whether the sample is contaminated. If there is significant and consistent amplification in the negative control, that batch of samples should be removed from the process and each stage should be evaluated for possible sources of contamination. Negative controls can also aid in determining the limit of detection. Examples of commonly used negative controls include DI water, a no template control, a no RT control in the case of assessing reverse transcription, and samples containing no detectable concentrations of the target of interest. Positive controls can be added at any stage in the process to compare differences in recovery between methods and to determine the recovery of the target or a similar target throughout the process. Positive controls are also commonly used during the PCR amplification stage to validate positive wastewater detections. Negative controls are in large part used to track contamination throughout the process and identify false positives, as well as aid in determining the limit of detection. To wrap up the process control discussion, I'm going to briefly give an example of how internal standards were used to identify a sampler issue in our lab. In the spring of 2022, we noticed that we were getting inconsistent SARS-CoV-2 concentrations in routine samples collected from one of our wastewater treatment plants. In addition to SARS-CoV-2, we measure PMMOV in these samples. PMMOV is a positive external standard and is commonly used in environmental samples as a human fecal indicator because it is shed in feces at high levels. By measuring PMMOV, we can confirm that we are collecting representative samples that include inputs of human fecal matter. It turns out that there was, in fact, little to no PMMOV present in the samples collected from the wastewater treatment plant in question as compared to the other sites. We then notified the other Houston WBE facilities of the issue in the health department as well as Houston Water were able to determine that the auto sampler was not functioning properly. The sampler was then repaired, and the results for that treatment plant were in line with what we were seeing across other sites. Lastly, I'm going to go over factors that impact microbial recovery in wastewater samples, ways to impact recovery, followed by an example from our lab. Microbial loss can occur anywhere in the process for various reasons, including improper storage conditions, exposure to enzymes that destroy nucleic acids such as DNAs and RNAs, inhibitors either from the sample matrix or carryover from other stages in the process, incomplete nucleic acid reactions, and the morphology and partitioning behavior of the microbe in wastewater. As previously mentioned, human fecal markers, surrogate microbes, and the target microbe can be used to estimate recovery throughout each process. Recovery can be improved by storing samples at the appropriate temperature. This might be 4 degrees Celsius if being processed within 24 hours, and minus 20 or 80 degrees Celsius for long-term storage. You would also need to minimize freeze-thaw cycles to prevent sample degradation. And if possible, you want to process samples immediately or within 24 hours of collection. Decontaminants can also be used to eliminate enzymes on working surfaces that could possibly destroy your target microbe. Another way for improving recovery is to determine the partitioning behavior of your target. There are a few published studies that show influenza A, for example, to partition more to wastewater solids as opposed to the supernatant. Bearing in mind that recovery not only depends on the microbe, but also the sample composition and the sample processing methods. It is good practice to perform these experiments on your own samples in order to assess which sample type will give you the best recovery. Inhibitors are substances that can interfere with PCRs and prevent target amplification. As previously mentioned, they can be found in the sample matrix or introduced throughout the process. Dilution, the addition of inhibitor-reducing reagents, and choice of concentration and extraction methods can all help to reduce inhibition and improve recovery. For this last example, I'm using some results collected from a partitioning study conducted in our lab. As I mentioned before, there are a few studies that determine influenza A to partition more to the solids fraction than the supernatant. In order to test this in our own samples, we obtained a sample of wastewater, placed a predetermined volume aside as a negative control, and spiked the remaining sample with influenza A as a positive external standard. The sample was homogenized and allowed to equilibrate before being aliquoted and centrifuged for another 30 minutes. This supernatant was then decanted and concentrated as usual while the leftover solids from each sample were resuspended using a lysis buffer and directly extracted. I also want to add that the solids for each sample were weighed throughout the process. Recovery was then calculated using the equation seen here, with copies per milliliter of wastewater being the concentration in the sample and copies per milliliter of wastewater of spiked virus being the concentration of the standard that was spiked into the sample. Since there was no amplification of influenza A in negative control samples, we assume that the concentrations seen here are reflective of the virus that was spiked in. The graph shows the replicate samples on the x-axis, the concentration on the primary y-axis, and the percent recovery on the secondary y-axis, where the dotted lines represent the trend of percent recovery across the 3 replicates for each sample type. Our results suggest that we recovered slightly higher concentrations of the virus in the supernatant of our samples than the solid fraction. For this reason, we decided to assay the supernatant to quantify influenza in our routine analysis. In summary, routine cleaning of equipment and workstations, separating work areas, and implementing automation where possible are several measures that can be taken to reduce contamination and ensure accuracy of results. Positive and negative controls are critical for validating results. They function to determine whether sample contamination is present and whether the quantification process is working. They can also be used to estimate target recovery and establish the process limit of detection. Target loss can occur at any stage in the process for various reasons, including improper storage, presence of inhibitors, and target morphology and partitioning behavior. Storing samples at the appropriate temperature, immediate processing, utilizing surface decontaminants, and minimizing inhibition are a few measures that can be taken to reduce target loss in wastewater samples. If you're interested in learning more about sampling, analyzing, and integrating wastewater data into public health interventions, check out our other training videos on our website. Support for this program was provided through a grant by the CDC. The Houston Wastewater Epidemiology team would like to thank you for viewing this training video.